So we're going to begin talking about oxidative phosphorylation, but first we're going to talk about the mitochondria, where the oxidative phosphorylation occurs, and we'll discuss the structure and the function of the actual mitochondria. So the objectives here are to know how the structure correlates to the function of the mitochondria and the different various compartments, what they contribute to energy production, as well as knowing the location of the components of the electron transport chain specifically. In addition, we'll talk about how things get in and out of the mitochondria. So the mitochondria itself is a oxygen-consuming organelle, which is free floating throughout the cell. Mitochondria are known as the powerhouse of the cell since the enzymatic oxidation of different substrates releases energy. They are going to be abundantly found in sites where energy is needed, such as muscle cells, liver cells. Liver can have up to 1,600 mitochondria. An oocyte can have about 300,000 mitochondria. In a general cell, you're going to have about at least 2,000 mitochondria, and this is going to be about 25% of the actual cell's total volume. Now here we have a picture of a mitochondria on the left, and we also have a picture of a bacterial membrane on the right. Mitochondria are remnants of a bacteria, and you can see within the membrane of the bacteria are going to be the electron transport chain. Bacteria do not have membrane-bound organelles, so the only membrane they have is their cell membrane. And so the electron transport chain will be incorporated into the membrane of the mitochondria. Now, the bacteria that are gram-negative have an outer membrane, and they have an inner membrane, and the electron transport chain is on that inner membrane. Our mitochondria look similar to a gram-negative bacteria, and the mitochondria have 4 important structures associated with them, and these are going to be the outer membrane structure, the inner membrane structure, the matrix, and then also the inner membrane space. Now, the inner membrane space is going to be where protons are going to be pumped against a gradient, and that's going to generate the potential to make ATP. Now, the inner membrane space is shown a bit better in the bacteria over here, in which you have hydrogens that are being pumped across the membrane, the inner membrane, into that inner membrane space. And then that's going to generate a potential to create ATP, and it's going to be very similar in our cells as well, in our mitochondria. Now, the outer mitochondrial membrane is going to be associated with the plasma membrane, or look more structurally similar to a plasma membrane. There are going to be porons in the outer membrane, and those are going to be more similar to our gram-negative bacteria. They're going to be beta barrels that make these pores. The inner mitochondrial membrane is going to have many enzymes, coenzymes, and different substances associated with the electron transport chain. You're also going to have proton pumps. The inner membrane is going to have outgrowths that are cristae, and these are going to be folds in the mitochondria inner membrane, these folds. The inner membrane is also going to be impermeable to many molecules. Gases, however, can cross the inner membrane, such as carbon dioxide and oxygen, and there are going to be numerous transporters for the inner membrane, and those transporters are important, such as ATP. ATP is going to be made in the matrix, and ATP has to be released to the cytoplasm for the cell to use it. So ATP has to be able to cross the membrane, for example. And the mitochondrial matrix, of course, is where you're also going to have the Krebs cycle, which we talked about, the citric acid cycle. Now, this is an example of the inner membrane of the mitochondria. And so you can see that embedded in the inner membrane are going to be enzymes, and these enzymes are going to be the electron transport chain. You're going to have a potential with these hydrogen ions pumped out of the matrix into the inner membrane space. And these are eventually going to flow back into the matrix through the ATP synthase, and this is going to generate what's very important, which is ATP. So the electron transport chain is basically transferring electrons in order to pump protons into the inner membrane space to create a potential, electrochemical potential, to drive the synthesis of ATP. Now, the inner mitochondrial membrane is going to be impermeable, again, to many things. And so if you have to have something that needs to go in and out of the matrix, you need to have a transporter for that. And so we're going to have different translocases that are going to transport polar molecules. We're also going to have an adenine nucleotide translocase, or the ANT, and that's on the inner mitochondrial membrane. And this is going to exchange ATP and ADP, and it's a one-to-one anti-port exchange. We're also going to have a symport with hydrogen and phosphate. We're going to have a symport with hydrogen and pyruvate. And we're also going to have the ability for calcium to enter. So there's going to be a calcium transporter. Now, across the inner membrane, of course, we have the electron transport chain that will pump hydrogens out, and we'll have the ATP synthase embedded, which will generate ATP. Now, there are other transporters on the membrane. Some we've already discussed. We have a few carboxylate transporters. So a dicarboxylate transporter is a phosphate malate exchange. We have a tricarboxylate transporter, and this is a citrate malate exchange. There's the aspartate glutamate transporter, which is part of the malate aspartate shuttle, and then a malate alpha ketoglutarate transporter. And so these are going to be important when we have too much energy to move carbons from the matrix into the cell, other areas of the cell where they can be used for other things. Now, the outer membrane, again, is going to resemble more of a bacteria. And so in your outer membrane, you're going to have nonspecific pores. And so these nonspecific pores are going to form what's called a voltage-dependent anion channel, or VDAC. And so if you have a compound that's 6,000 Daltons or less, it can typically pass through these voltage-dependent anion channels. And these are going to be, again, those beta barrels, which are very common in bacteria that form these sort of pores in the outer membrane. Now, another thing to keep in mind is that the mitochondria, since it's generating energy, ATP, many kinases that use ATP to phosphorylate, for example, are going to associate themselves with the mitochondria outer membrane on the cytosolic side. So as soon as ATP leaves that ANT transporter, antiport, and ATP gets out through then the voltage-dependent anion channels, those kinases are there to use that ATP for their purposes. Now, there is another type of pore that can form. It's called the Mitochondria Permeability Transition Pore. And in this situation, this is typically going to be during pathological conditions, stroke, brain trauma, when a cell is undergoing apoptosis. And the opening of this mitochondrial permeability transition pore will lead to apoptosis or cell death. And this is a very regulated process. And so basically you have the inner mitochondrial membrane where we had that adenosine nucleotide translocase, which was an antiport for ADP and ATP. This is now going to merge with the voltage-dependent anion channel, VDAC, which is typically that barrel on the outer membrane, and they're going to form a much larger pore. And this is going to allow hydrogen ions, cations to flood into the mitochondria. And the mitochondria is going to swell because water will follow to neutralize those anions and cations. And then this is going to cause the mitochondria to have irreversible damage and initiate or help to initiate the caspase cascade for apoptosis.